Guys, it is the last sprint. It is the home run. It is mind over matter at this point. Welcome to the last minute advice for A-level biology paper 3 exam. And yeah, it's a general consensus amongst everyone that paper 3 is the academic comeback of the century. Like, it doesn't matter if you've done good or bad. Like, I was thinking to myself that, yep, paper 3 is going to be my paper. I'm going to own it different because it's the last one, right? It's just like, let's just seal the deal in the most positive and the best way. And so, you have the strength to make it happen. So make it happen. As always, got 3 stars last year. I remember what I've done. You know, take it or leave it. But I was going to say, I did do OCRA. And honestly, this video is completely applied to those people who take that exam board. But if you take another one, take my advice with a pinch of salt. Because I know you AQA people have like a 25 marker, like the essay, right? Yeah, don't ask me for advice on that or tips. I haven't done it, so I can't say. But yeah, please just take this video with a pinch of salt if you don't take OCRA. But still, you know, I'm going to go through exam technique and stuff. It might be beneficial for you. Okay, as always, I have a total of 3 steps. So number 1, practicals are solidified in your head. Step 2, exam technique, which I'll go through. And step 3, past papers, and then last, run off the content. Right, practicals. Okay, paper 3 is known as the unified paper for a reason, because, you know, unity, no, I'm joking. It's bringing all the modules together, and they focus a lot on practicals and practical skills. So not only do I need you to know all your practicals, but I want you to have like a criteria in which you're assessing whether you actually know it or not. So like, are you able to explain the method to someone? Like, act as a teacher and say it out loud. Can you tell me points of validity, the control variables, independent, dependent, you know? And also, these are the things we're going to write in those like 6 markers on, like, oh my god, outline a valid experiment or method to do X, Y, and Z. Like, these are the things you would say. So be able to identify them from the practicals themselves. It's just, you're going to really develop your skills in that way for when exam questions do come up, which I do have some in this video, when they're like, oh my god, just tell me some, like, 2 improvements. Yeah, it's just priming your brain beforehand, and then when you get the exam questions and practicals, it'll be easy for you. YouTubers go through, like, the content aspects of the practicals, but I'd also do something else as well. Let's say it was about dissection of the, I don't know, the heart. Search that onto YouTube and look at somebody actually doing the practical. Most likely it's going to be a teacher in a lab, right? Watch it. Try and understand each step, okay? And not going to lie, when they do the practical, they say like, oh, I'm doing step this because of this. And those are because of are very important because they come up in the exam most likely. So please do pay attention to that. And honestly, visualizing and being able to see the practical is much better than reading off like a textbook. Yeah, no, I'm not doing that. Yeah, so that's that, practicals, okay? Step 2, exam technique. As always, my personal method is like, read the question first, not the information, because I get confused. And my answer is like a whole 180 away from the answer that's on the mark scheme, so I've learned that I shouldn't do that. Secondly, I'd rephrase the question, check the command word, because, guys, the questions, right? A question on outline or describe are not the same. A question on compare or explain are not the same. You can't treat them the same way. And I've been preaching this since the moment I've started this channel in October last year. Like, they are not the same, so don't treat it in that way. Again, if you need help on that, I've got a whole playlist. I've got tons and tons of walkthrough videos on different topics, different exam questions. So go have a look. And I basically really go through my method. Then after checking the command word, I see whether I actually need the information or not. Most likely I don't, you know, but the times that I do, I must use it because I can't answer it directly as in the question, and I just answer it lastly. And if you guys watched my paper 2 video, which I'm assuming you have, I hope you have, gosh, you would understand this reference about Nick Wilde, yeah? Let's just be very cunning and very slick about answering the questions, right? Stating the obvious, bish bash bosh, get it over and done with, call it a day, yeah? And you'll see what I mean as I go through the questions now. So, okay, I'm going to read the question first. So, suggest how using gene therapy to increase levels of AC6 gene in heart cells may improve heart function. Yeah, I have no idea what's going on, so I'm going to read the information. So, gene therapy can do that. Now, this is the important part. Okay, the gene codes for one form of the enzyme adenylyl cyclase. Okay, and they've done clinical trials, and it's caused an increase in those levels. Now let's just go back to the question. As I said, this is the important part. This line, so irrelevant. I don't really care. Let's go back. So it's saying, if I were to rephrase it, if we've increased this gene, how is it going to affect the heart cells? And I circled cells because for like an enzyme or hormone or something to affect a cell, we're talking about the plasma membrane here. Okay, right now I'm like bringing in many topics together, trying to piece an answer as well. So we're going to state the obvious. If you're increasing that gene, well, hello, you're going to increase the amount of adenylyl cyclase. First mark. Secondly, well, how is it going to affect those cells? Well, it's going to bind to the plasma membrane. That in itself is 2 marks. I haven't even said what's actually happening, right? Well, I mean, we can take it a step further, be like, yeah, there'll be more CAMP, secondary messenger, because inside the cell, that then causes the cell activity to change, and therefore would improve the heart function, which in this case would be, I don't know, contraction of cardiac muscle. But they're just extra points. Honestly, let's be real, everybody can get these first 2 points. It's just stating the obvious. Also, there's, that question about nerves, and it's like, oh, we've blocked this channel with this drug. What effect does it have? We're talking about nerves here, right? Just say the calcium ion channels, if they're blocked, that means calcium ions can't enter. Do you get it? Like, just say it. So here, more adenylyl cyclase on the plasma membrane causes the effect. Easy 2 marks. Okay, and this is a suggest question, which, to be honest with you, they're a bit hit or miss sometimes. Most of the time they're a hit, but it's a bit, sometimes they're a bit of a miss. But here, it's very simple, isn't it? Just derive what you need and go from there. Now, describe and explain how the student's procedure could be improved. Okay, I don't know what the procedure is, so let's read it. So I'd always identify what is it they're doing in the first place. So they're cloning a plant, yeah? Okay, select a stem with many flowers and leaves. No. Alarm bells are ringing. No, you don't want to do that. I'll explain later, but already I'm thinking about why this is problematic. I haven't said it yet, but as you go through bullet points, just think, is this good or bad? How can it be improved? Next, make a slanting cut, dip the cut stem into rooting powder, plant the cutting in watered compost. Okay, whenever you're cloning something, especially a plant, you want aseptic techniques. There is no mention of that. Okay, also, you don't want many leaves at all, because then that leads to transpiration, does it not? That's why I said no. You don't want that. So already we've got 2 points, right? So if we look over here to the mark scheme, it says use aseptic techniques because you don't want contamination. The moment one plant, or that plant you're trying to clone, is contaminated, well, then the rest of your plants are contaminated. What's the point, You just defeated the whole purpose because they're not going to be healthy. So you just wasted energy and time and resources. So that's an important point. You can always chuck in. Secondly, remove most of the leaves because you don't want there to be loss of water, right, through transpiration. Easy. Other things might be use a greenhouse because you want to control the environment in which it grows in, and you also want optimal temperature, moisture and stuff like that because it's a plant. They've got requirements, they've got needs. So the method here really is just think, okay, the purpose of this whole method is to clone. What conditions does this plant need? Have they said it? No? Okay, say that as an improvement. Okay, but this gets easier once you know the practicals, right? Or when you read through stuff about cloning. So that's how you do that. What's this next question? Okay. Using the graph, describe the conclusions that can be drawn about the role of different oxygen concentrations in the control of apical dominance. Gosh, that's too many words. If I had to rephrase it, I would be like, yeah, use the graph 100%. Okay, you probably won't get any marks if you don't. Secondly, if it's conclusions, especially describe the conclusions, I know for a fact I'm going to be like, girl, state the obvious, say how it is, don't try and explain things. Just try and say what's going on the graph. And then I would just say like, what effect is it having on apical dominance? So if we go here, we can see that the graph is quite interesting. We've got negative and we've got positive on the y-axis. Stimulation of growth, as in it's growing, and inhibition of growth is not really growing. I've kind of drawn like, not really line of best fit, but some lines, just so that I can see the pattern almost. And from here, we can see that, okay, at higher oxygen concentration, remember, this is lower, this will be higher, of course, because it's going to go increasing, but just in case if you ever get confused with like standard form and stuff, right? At higher levels of oxygen concentration, there is apical growth or apical dominance because it is at the positive region, as in like positive like y-axis, the percentage is higher. Okay, that's easy. I just said how it is, right? Also, if it's a describe the conclusions and it's a graph, always say where it peaks. So here it peaks at like 10 to the power minus 5 moles per decimeter cubed. It's kind of like describing the graph, but it's also like link it to whether it's growing or not, sort of vibes. Next thing we could probably say is that, oh, at lower oxygen concentrations, right? What is happening here? I would say that yeah, the growth has been like it's increasing because it's, you know, this is 0, right? If it was below here, then you would say that, yeah, like it's been inhibited, but because it's positive, it means that there is basically apical dominance because of the stimulation of growth. So we can say that at lower oxygen concentrations, okay? So with this, you kind of want to split the graph into like 3 parts. One, the lower end, second part, the upper end, and third part, like any significant things on the graph, so like a peak, yeah? So that's basically how you do that question. And then this, get me any question with practicals, like especially a 6 marker. Like, this is my thing. I love it so much because it's the same thing every time. Basically, you guys need to know that if you get, oh my god, outline the method, you're literally going to say the same thing every time. You're going to identify the independent, so say what it is. You're going to say the dependent variable, also how you are going to measure it. I would say list like at least, I say 3 controls. Also to say how you are going to control it. Are going to control it, or them. Next thing, really method, like, you know, what is the method? Remember, you don't need to go that specific. Just imagine, right? You were in school, you had to do this practical. The steps aren't going to be, you know, complicated. It's just going to be very methodical. So just try and think methodically in that way. Of course, by doing step 1, going through practicals, you would already be thinking in that methodical manner because you've been exposed to all these different methods. That's why I put that as step 1. Method and then points of validity. So I can literally recite this in my sleep. Repeat, calculate a mean, identify anomalous results, standard deviation, stats, la 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 la. You get the point, right? This is like your framework of what you're going to do for these sort of questions. If we were to look at the mark scheme, basically lines up. Also, always mention a control group. Okay, not many people, no, I'm overgeneralizing here, but some people don't know what a control group is. It's just like a group that it's not really affected. Like, let's say the practicals, as I said here, affect gibberellin concentration or stem elongation, right? One group would just not have any gibberellin at all. That would be classed as the control group, and you always want to identify that, whether the practical has it or not. And if it doesn't, that's an improvement, just so you know. Yeah, okay. Next one. Outline 2 ways in which the thermoregulation in naked mole rats is different in the thermoregulation in most mammals. Admittedly, there was a photo here, but just know that they don't have any fur and that it's basically skin. Okay, that's just how they look. You probably have seen a photo of them because OCR loves it. I don't know why they love this mammal, but sure. If I were to rephrase it, they're saying, yep, thermoregulation in these rats is different to most mammals. So think about humans, because we are mammals, right? So humans, we're at like 37 degrees Celsius. Okay, we've got basically our thermoregulations essentially on physiological changes. What else? I'd say, we have hair, so that also affects it. So I've already just primed my brain into thinking what other mammals have. Now, when I read through these bullet points, I'll be able to be like, that doesn't add up, and that would be the answer, So, right, they live in complex underground tunnel systems, and to have a stable temperature of 30 to 32 degrees Celsius, right? Immediately, that doesn't add up to the 37 degrees Celsius that I was talking about. So that's 1 point. Secondly, you can see that, temperature of the tunnel increases, they move to a cooler part. Oh, if it decreases, they often lie together. That is classed as behavioural responses and changes and whatever. So, that's different to our physiological responses, right? Because what do? We sweat, we shiver, we vasodilate, vasoconstrict. Like, we do all of this. We're not cuddling together. What's going on there? So yeah, this is class as behavioural, and we could say that. We could be like, yeah, they have more behavioural responses or fewer physiological responses, and that lines up with the mark scheme. Just say it how it is. I hope you can tell by my nonchalant manner of answering this question, like, it kind of proves the point. I hope so. That's the goal, but yeah, just say it how it is, okay? Remember, be like Nick Wilde, yeah? He's such a goat, I swear to God. Next one. Explain how positive feedback could accelerate the process of hypothermia. I'll be so honest with you guys. I remember when I did this question in year 13, I think I only got 1 mark. And I was so mad. I was like, why? What have I done? And then I realised, this is quite interesting, how I'm going to approach this question. Because firstly, it says explain, yeah? Explain means, say how it is, content, regurgitation of content. But I just didn't know what to regurgitate. So that 1 mark that I did get was defining what positive feedback is. And not gonna lie, if a question has like a key term or keyword or whatever, define it, most likely a mark. But the interesting part is the information they gave above, which was, in humans, when core body temperature falls below 35 degrees Celsius, positive feedback causes this decrease in core body temperature to continue. Technically speaking, they're saying that, yep, temperature keeps decreasing. Now the question is saying, how is positive feedback leading to this? Right? Remember, there is an association, okay? When you think temperature, you think enzymes. They go hand in hand. They are brother and sister. They are like best friends. And that's what I learned from this question when I first did it. I was like, whoa, this is crazy. There's no way. So, positive feedback basically is when a change is further exaggerated. So here, temperature is decreasing. Positive feedback's like, yep, let's make it worse. Let's decrease it even further. But because this is explained, it's content, right? So what content are we relating it to? Well, we think about enzymes. Da-da! So here, if there's lower body temperature, lower rate of, or basically reduces kinetic energy, enzyme activity would reduce. Respiration rate would reduce. Metabolic processes would reduce and whatever. And also, this is actually baffled me. I can't even lie. When I first did it, I was not on this level. Less metabolic processes means that there's less heat generated. Let's just take a moment for that. If you knew that, I respect you so highly. And if you don't, I also respect you because I didn't know. So you learn so much from the mark schemes, to be honest. It makes sense, right? If there's lower rate of metabolic processes, there's less heat. That would lead you, for the body temperature to decrease even further. And that's crazy. This is a whole 1 mark in itself. temperature drops further. Like, come on, guys, say it how it is, you know? So that's that question. Then another practical question is this. Suggest and explain 2 improvements that would increase the validity of the student's investigation. I'm always thinking about certain things. 1, control variables, 2, control group, and 3, the intervals at which they're measuring that thing in, right? Here is temperature. 10, 20, 30, 40, 50. Yeah, mate, that's too large. Like, the gaps are too large. What if the optimal was like at 12? How would you know? You wouldn't. So that's why that would be an improvement, you know, use smaller intervals of temperature so you can get the optimal temperature needed for this enzyme. As I said, I'm looking at control variables. They haven't really said anything, you know? It's needed. So it's like you can standardize the method, right? Because if you can't standardize a method, well, that practical's basically gone out the door because you can't apply it to other situations. And again, no mention of control group. Yeah. So that's the mark scheme for that, okay? So that's the exam technique aspect of it. And step 3, past papers. I need you, like, I don't even care at this point how many you've done. I don't care if you need to redo them. Keep redoing them. Keep doing the past papers, because the thing is, right, paper 3 for OCR is quite, like, it's a fun one. I actually really enjoyed it. Like, it's a mix of everything, is it not? Thank God it didn't have multiple choice. I don't know, I just don't like it, but it's got practicals, it's got like 6 markers, it's got explain, it's got evaluate, it's got everything, you know? Love it. So, and also, the questions are much more straightforward. Listen, that's my personal opinion. I just think they're more straightforward than like paper 1 and paper 2. So you need to get into the manner of it, you need to get into the swing and the swing of it, really. So definitely speed run some past papers, and while you do them, make a list of things that you spot. Like, for example, if it's an evaluate question, you say X, Y, and Z. If there's practical questions, you write whatever. So these are like 2 post-it notes that I had made based on things I'd observed, you know? Because paper 3 is assessing all the content, right? So that's why I also say, do your last run of the content. Because I know loads of you did all-nighters on before paper 2. You guys told me, I know. Because it was a last minute thing, you probably have forgotten the content. So definitely please review your content as well. Because as I said, right, paper 3 questions are much more straightforward. Yeah? There is application, but there is a lot of content questions as well. So it'll be a shame if you don't get the content questions in the first place. So, yeah. And always, if you're doing a little mini notebook, you get brownie points. If you get the reference. And lastly, gosh, fight for the marks. Paper 3 comeback is incoming. We're gonna do it. Yeah, it's gonna happen. Just keep on studying, keep on grinding, okay? You've got what? 1 week left? Pfft, please. How many months do you have for holidays? Yeah, the sacrifice is justified. Just study until you can't get it wrong, as Rory says. And please be as calm as possible. The best performers are the ones who are calm, okay? And I guess it's kind of like the last video, almost. I really wanted to say thank you so much for all your love and support. I genuinely had no idea this channel was going to grow this fast. It actually baffles me till this day. Yeah, no, it's been so great, and I love the community, like in the comment section. It's actually such vibes, and it makes me laugh so much, you guys. You guys say such out-of-pocket things that I love. And I just wanted to say that I really do hope all these videos were beneficial for you. And I hope that, at the end of the day, on results day, we'll speak before that, I'll post a video for results day, but I just hope you get the grade that you want. I hope you can get, go to the next step that you want to do in life because of your grades. And yeah, that's all I have to say, really. Just go smash it, man. It's the last leg. That's it. Just do it. Honestly, just do it. Because if you don't do it now, when are you going to do it?